This video describes our central findings in the following study. Prostate cancer is responsible for more than 34,000 deaths each year in the US alone. Prostate tumor biology is driven by the androgen receptor, and for this reason, advanced tumors are treated with therapies targeting the AR signaling pathway. Unfortunately, nearly all advanced tumors will eventually recur, and so there's a major unmet need to understand how tumor cells survive AR inhibition in order to develop new strategies to prevent or delay prostate cancer progression and lethality. We know that metabolism can be reprogrammed during tumor progression and treatment resistance in order to fuel the changing demands of tumor cells. Targeting metabolism has shown promise in models of aggressive prostate cancer. Stimulation of the androgen receptor has been shown to promote certain metabolic processes. And so we hypothesize that understanding how metabolism is reprogrammed in prostate cancer cells that survive AR inhibition may yield new adaptive mechanisms and therapeutic opportunities. The goals of this study are first to define AR inhibition-induced metabolic reprogramming by integrating transcriptional profiling, metabolic profiling, and bioenergetics across clinical tissues, patient-derived xenografts, and cell lines. Secondly, to identify treatment-induced metabolic pathways that support tumor cell fitness and may represent therapeutic vulnerabilities in the cells that survive AR inhibition. We grew tumors in mice, treated a subset with the AR inhibitor enzalutamide, and performed metabolic profiling, identifying metabolites that are enriched or depleted after AR inhibition in vivo. To explore bioenergetics, we measured oxygen consumption rate, OCR, and extracellular acidification rate, ECAR. We found that the glycolytic ATP production rate was dramatically reduced in enzalutamide-treated cells, while the mitochondrial ATP production rate was not altered. This means that oxidative phosphorylation comprises a greater percentage of the overall ATP supply after AR inhibition. As a complementary approach, we performed isotope tracing with labeled glucose, finding less enrichment into lactate in ENZA-treated cells, whereas enrichment into TCA cycle intermediates was largely maintained. Our data support a model whereby AR inhibition leads to reduced glycolysis, but maintenance of oxidative mitochondrial metabolism. So we asked, what is responsible for reduced glycolytic activity after AR inhibition? In cells treated in vitro or in vivo, including PDX models, we found a consistent reduction in expression of the glycolytic enzyme hexokinase 2 after AR inhibition. We validated reduced HK2 protein levels using immunohistochemistry. HK2 RNA levels are also reduced in patients treated with androgen deprivation therapy or in metastatic castration-resistant prostate cancer treated with enzalutamide. We found reduced MYC signaling with enzalutamide, and restoring MYC was sufficient to partially rescue HK2 expression. Targeted bisulfite sequencing identified increased CpG methylation at the transcriptional start site of HK2 in enza-treated cells, suggesting that multiple mechanisms may contribute to reducing HK2. To understand the consequences of low HK2, we knocked it down and measured ECAR after treatment with the ATP synthase inhibitor oligomycin, which reduces OXPHOS and stimulates a twofold increase in ECAR of scrambled control cells. However, the rate was unchanged in HK2 knockdown cells, suggesting that low HK2 contributes to reduced glycolytic capacity. As cell stress can induce changes in mitochondrial morphology, we stained for the mitochondrial protein TUFM. Immunofluorescence identified robustly elongated mitochondria in ENZA-treated cells, which we quantified by measuring aspect ratio and form factor. We believe mitochondrial elongation is a consequence of reduced expression and phosphorylation at serine 616 and DRP1, a driver of mitochondrial fission. We asked if we could rescue mitochondrial fission, and if so, how this might influence cell fitness. We expressed a phosphomimetic version of DRP1, which was sufficient to promote mitochondrial fission, validated by reduced mitochondrial aspect ratio. In ENZA-treated cells, this led to increased apoptosis and reduced proliferation using an EDU labeling assay. These phenotypes were specific to the ENZA-treated cells, suggesting that mitochondrial elongation may benefit cells that survive AR inhibition. Finally, we asked if we could target oxidative mitochondrial metabolism after AR inhibition. The complex 1 inhibitor IAX-10759 potently suppresses respiration. After first treating PDX tumors with a vehicle or enzalutamide, we found that IAX had the greatest reduction in proliferation on ENZA-treated tumors, and we found a statistically significant reduction in growth with IAX only in ENZA-treated tumors. We also did the flip side of the experiment, pre-treating cells with IAX or vehicle and then evaluating response to enzalutamide. The greatest response came from cells pre-treated with IAX followed by enzalutamide. And proportionately, IX pretreatment effectively doubled the response to ENZA. In summary, we find that AR inhibition dampens glycolytic activity through reduced HK2 expression. Mitochondrial elongation through impaired DRP1 expression and activity, promotes survival and growth of tumor cells after AR inhibition. Cells that survive AR blockade are sensitive to inhibition of oxidative metabolism, and we believe that defining treatment-induced reprogramming may yield treatment-associated metabolic vulnerabilities. In the context of clinical prostate cancer, inhibition of oxidative metabolism in combination with AR inhibition may reduce tumor cell survival and proliferation in order to delay or prevent disease progression. Clinical trials with metformin for prostate cancer have been inconclusive. However, metformin is known to have a number of targets in addition to oxidative metabolism, and it is unclear if the concentration of metformin that reaches tumor tissues is high enough to inhibit oxidative metabolism in vivo. Therefore, there's a need for better clinically viable inhibitors of oxidative metabolism in order to test our hypothesis in prostate cancer. We want to thank the co-authors, collaborators, and funding sources who supported this research, and we look forward to learning more as we continue our studies. Thank you for watching.